with us today. Okay. So the diagram that you're looking at is a familiar diagram that I've shared on a couple of times. So this is just a summary that is the diagram that is summarizing the function of the kidney nephron and also the structure of the kidney nephron. So it gets more complicated than what you're looking at right now. You know to say that on one end you have the glomerulus. The glomerulus is the one that is filtering substances from the blood. So in the blood you have the glomerular capillaries that are intertwined within the glomerulus. So those are the ones that are responsible for filtration. So there are substances that are being filtered there by the glomerulus into the Bowman's capsule or Bowman's space. Once those filtrate gets into the Bowman's space, they'll be transported or delivered by the kidney nephron. So this is the kidney nephron. We have different segments of the kidney nephron, starting with the proximal tubule. So you have the convoluted proximal tubule. Within the convoluted proximal tubule, you can see that there are proteins that are involved in reabsorption and secretion. So you find that there are certain things that were filtered, but they are still needed by the body, so they will be reabsorbed. So within the proximal convoluted tubule, you have those protein pumps, channels that are responsible for reabsorption. So certain things will be reabsorbed. For instance, glucose, amino acids, some of the electrolytes that needs to be reabsorbed back into the body, they will be reabsorbed here. Then there are certain substances that were not filtered by the glomerulus, but the body needs to get rid of them. So you find that they can be secreted by the tubular capillaries. So you have the tubular capillaries that are responsible for secretion of certain substances into the lumen of the kidney nephron. So you can see that secretion taking place. Then as you go down, you have the loop of Henle, where you have absorption and secretion of other substances. So you have reabsorption of water. For instance, you can see there's osmosis taking place within the descending limb or the loop of Henle. The ascending limb, you have reabsorption of salts. So you can have salt pumps that are responsible for reabsorption of salts. And these salts, mainly sodium, chloride, you have magnesium, and you have potassium, a lot of these electrolytes that needs to be reabsorbed. Then as you are going towards the distal convoluted tubule, you can see there are also channels that are responsible for reabsorption and secretion, just like what we saw in the proximal tubule. So each and every segment is responsible for a particular function that we are going to be discussing. So this is a kidney nephron. So this is more of a summarized diagram, but we'll go into detail to look at what is being reabsorbed and secreted at which point of the kidney nephron. So before we proceed with those functions, it's very important that you understand the anatomical aspects of the kidney nephron, so that as we are discussing the physiology, you already know the anatomy. So the structure is very simple. I think you've discussed this in other courses like anatomy, maybe pharmacology to some extent. You can also discuss this, and also other courses, prerequisite courses that you had before you started your program, your nursing program. So this is something which is not strange to a student. You are familiar with this structure. So you can see that you have the afferent arterioles that are feeding the glomerulus with that blood. So they're going to deliver blood to the glomerulus via the afferent arterioles. Then from there, they're going to divide to form the capillaries. So you have the capillaries that are responsible for filtration, and then those capillaries can join to form the efferent arteriole. So the efferent arteriole later on becomes the peritubular capillaries and the Vasalacta. I think that kind of organization we already discussed when we are looking at the functional anatomy of the kidneys. So after the glomerulus, we have the Bowman's capsule. The Bowman's capsule is just a membrane that is covering the glomerulus. So you have the membrane that is covering the glomerulus is called the Bowman's capsule. Then within the Bowman's capsule, that's where you're going to find the Bowman's space. And the function of the Bowman's space is to receive the filtrate. So the ultrafiltrate, these are the fluids and electrolytes, nutrients that are being filtered. Then they'll become ultrafiltrate. and that ultrafiltrate is received by the Bowman space. Then from the Bowman space, it's connected to the proximal convoluted tubule. So you can see the proximal convoluted tubule. Then after the proximal convoluted tubule, we have the loop of Henle. So the loop of Henle is the hairpin bend structure. So you can see it resembles the hairpin. Okay, so ladies, they know what I'm talking about. So there are these hairpins that you use to clip your hair so that it's not very loose. So this is the loop of Henle that is also responsible for absorption and secretion of other substances, as we'll be discussing. So the loop of Henle is divided into descending and ascending. So descending is the one that is descending down, so it's going downwards. So you can see this is the descending limb of the loop of Henle. Then we also have the ascending limb of the loop of Henle. So descending and ascending. After the ascending, we have the distal convoluted tubule that is connected to the collecting tubules. And these collecting tubules, they are connecting to the collecting ducts. The collecting ducts will deliver the urine towards the papillae, and the papillae, they are connected to the minor calyces. Then the minor calyces will combine to form major calyces. The major calyces will combine to form renal pelvis. Renal pelvis will proceed as the ureter, and the ureter are connected to the urinary bladder. From the urinary bladder, you have the urethra that is delivering or maybe transporting urine to the outside world via the opening of the urethra. So you have the urethral opening that is responsible for removing of urine to the outside world during urination or menstruation. So this is just the anatomy of the kidney nephron. But in physiology, you're not just interested in the structure. You need to understand what is the function of the kidney nephron, what is it doing. So this is the major objective for today's lecture. So you need to understand. So the previous diagram and this one is almost the same. So you have the afferent arterioles that will form the capillaries within the glomerulus, and then they will proceed as the efferent arterioles. Later on, they'll become the tubular capillaries, and after that, they'll become the Vasalacta. So the Vasalacta are actually peritubular capillaries that are adjacent to the loop of Henle. So they are called Vasalacta. And then you have the nephron itself that is divided into the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, the collecting tubules, collecting ducts, just like that. So it's very easy. If I ask you to say, can you draw the kidney nephron? No one can fail to do that. But sometimes I'm very surprised with you students. I ask you very simple questions, but you fail to answer. So I don't know why. Whether you maybe just don't study or you're not paying attention whenever you're having a lecture. So it's so disappointing sometimes when you give a test expecting that students will perform very well, then you are disappointed. Okay, so let's start building up the information. So in this slide, we still have the anatomical structure of the kidney nephron, but I've also added a bit of information. So starting with one, we have the renal corpuscle. The renal corpuscle is the same glomerulus. So the renal corpuscle is a spherical structure consisting of the glomerular capsule, a cup-shaped chamber approximately 200 micrometers in diameter, and a capillary network, which are called glomerulus. So you have this cup-shaped structure within the kidneys, which is called the renal corpuscle. So this renal corpuscle is comprising of the glomerular capillaries, the glomerular capsule, and the glomerulus. So it's the one that is responsible for filtration in simple terms. So later on, we'll be discussing the membrane that is involved in filtration. We say that you have the endothelial cells that are coming from the capillaries, the renal capillaries. Then you have the podocytes on the other side. In between them, you have the basement membrane that is responsible for filtration. So I hope you remember that. Even when you're looking at factors that are affecting the glomerular filtration rate, we say that the permeability of this membrane is also a factor. The permeability, the charge, and everything. Okay, so after the renal corpuscle, we have the proximal convoluted tubule. So the proximal convoluted tubule is responsible for reabsorbing nutrients from the filtrate, now known as the tubular fluid. So once the blood has been filtered, so the plasma is the one that is being filtered here, because only these fluids are the ones that are being filtered. The cells will remain within the capillaries. So the plasma that has been filtered here is known as the tubular fluid, because now it's a fluid that is found within these tubules. So tubules are just like tiny tubes. That's why they're called tubules. So whatever has been filtered is called tubular fluid now. It will start moving within the kidney nephron. So the proximal convoluted tubule is responsible for reabsorbing some of the nutrients. For instance, glucose, it has to be reabsorbed here. Amino acids, they have to be reabsorbed here. Then some extent of electrolytes, for instance, bicarbonates, sodium, and other electrolytes, they have to be reabsorbed here. So it's mainly responsible for reabsorption of nutrients back into the body. Then after the proximal convoluted tubule, we say that we have the kidney nephron, and this is called the loop of Henle. So you can see you have the nephron loop. So this loop of Henle is responsible for the establishment of osmotic gradient in the renal medulla. So the osmotic gradient in the renal medulla is created in a phenomena which is called the countercurrent mechanisms. So later on, we'll be discussing the countercurrent mechanism. How does the loop of Henle come in to create the osmotic gradient that will facilitate the reabsorption of water, which is called osmosis. So all that is a function of the loop of Henle. So there is pumping of salts, there is also reabsorption of water. I'll explain that. Then after the loop of Henle, we have the distal convoluted tubule. The distal convoluted tubule is going to adjust the tubular fluid that is moving after the function of the proximal convoluted tubule in the loop of Henle. The distal convoluted tubule is also responsible for other reabsorption and secretion, so that you have the final production of the urine. So there is also secretion and reabsorption taking place within the distal convoluted tubule. Okay, and then we have the collecting system. So the collecting system, it's comprised of the collecting tubules and the collecting duct. So the collecting tubules that are connected to the collecting duct. So this is a system that is now responsible to deliver the urine that has been produced to the renal pelvis and later on to the urinary bladder. Okay, so you have the collecting system receives the urine from individual nephron and performs final adjustment in the urine volume and composition before delivering it to minor calyxes. So from the minor calyx, you have the major calyx. After the major calyx, you have the renal pelvis that is connected to the ureters. From the ureters, urinary bladder, then via the urethra and the urethral opening, the urine can be released to the outside world. So this is the collecting system. So mainly we have the collecting tubules and the collecting ducts. Okay, so the types of cells that are found in each segment are also different. So you have the thicker portions where you have more of cuboidal or cuboid cells. So you have the cuboidal cells, more like cubes. So you can see in certain segments of the kidney nephron, you have these cells that are more like cubes. These are epithelium cells. Then other cells are thin cells. So you find that you have the thin segment and the thick segment that we'll be discussing. Okay. Okay, so this diagram is also almost identical to the previous one, but here is specific the kind of things that are being reabsorbed and secreted by these segments. So the renal corpuscle is going to generate the glomerular filtrate composed by water, ions, and small molecules. So any nutrients that are smaller enough, they will be filtered by the glomerulus. Then they will move to the proximal convoluted tubule, where there is reabsorption of sodium, chloride, potassium, water, glucose, amino acids, bicarbonates, calcium, phosphates. Then there's also secretion of ammonium and creatinine. So ammonium and creatinine can be secreted by the proximal convoluted tubule. So here it sounds so simple because you have the information. But once I bring a question in MCQs or maybe a short answer questions, I ask you to say what is the function of the proximal convoluted tubule, you forget to mention this to say it's responsible for reabsorption of sodium, chloride, potassium, water, glucose, amino acids, bicarbonates, calcium, and phosphates. Is on top of that is also responsible for secretion of ammonium and creatinine. The same creatinine that can be used to estimate the glomerular filtration rate can also be secreted by the proximal convoluted tubule, which we said because some of it is being secreted here, it will bring about an error in the estimation of the glomerular filtration rate. I hope you remember that. Then after the proximal convoluted tubule, the loop of Henle, mainly the thin descending limb is responsible for reabsorption of water. Then the thick ascending limb or the ascending limb, the thin and thick, is responsible for reabsorption of sodium and chloride. That will create the hyperosmolarity of the renal medulla. So you can see the renal medulla, or this part of the kidneys, they are high in terms of osmolarity. The osmolarity is increased. Why? It's because in the thin ascending, so thin ascending and thick ascending, they are responsible for reabsorption of salts like sodium, chloride, ammonium, and sodium chloride in short. So you find that that is going to create the gradient, the osmotic gradient that will facilitate reabsorption of water in the thin descending limb. So this is involved in countercurrent mechanism that I'll be explaining in details later on. Then we have the distal convoluted tubule that is mainly responsible for reabsorption of sodium and chloride. The collecting ducts and the collecting tubules, they are also responsible for reabsorption of sodium, chloride, and water. Then they can secrete ammonia, so secretion of ammonia, hydrogen ions, and potassium. So this is just a summary, but now we need to go in details, starting with the proximal convoluted tubule, because we've already discussed the glomerular filtration rate. What is happening within the glomerulus, we have already discussed in the previous lectures, but now we need to proceed by looking at the nephron function, starting with the proximal convoluted tubule. So going in details now, let's understand the function of the proximal convoluted tubule. So what is the function of the proximal convoluted tubule? In such a way that if I ask you a question to say, maybe it's an essay question, explain the functions of the proximal convoluted tubule, you shouldn't just spend maybe half a page just to write that. You need to have a lot of information so that you can spend more time explaining about the functions of the proximal convoluted tubule. So it starts from here. You need to understand here. Okay? So if I'm very fast, you need to slow me down. If you're not getting what I'm saying, you need to slow me down. Otherwise, you'll be losing out. Nevertheless, we continue. So now let's look at the major functions of the proximal convoluted tubules. So the proximal convoluted tubule, this is the longest part of the nephron and is located in the cortex of the kidneys. Remember, the kidneys, they are divided into the cortex and the medulla at the center. So the proximal convoluted tubule is mainly located within the cortex. And we have two different types of kidney nephron, if you remember. We had the cortical nephron that are mainly located with the glomerulus is located in the cortex, and part of it is mainly located within the cortex. That's why it's called the cortical nephron. And then we also have the juxtamedullary nephrons, whereby the glomerulus is still located within the cortex, but the major part of this nephron is in the medulla. So I hope you remember those two different types of the kidney nephrons. So for both two types of kidney nephrons, the proximal convoluted tubule, they are mainly located within the cortex, Why are they located within the cortex? Is because the blood supply to the cortex is more as compared to the medulla of the kidneys. So if the glomerulus are the ones that are responsible for filtration, and then after filtration, they need to proceed to the proximal convoluted tubule. So you find that the glomerulus and the proximal convoluted tubule, they are located within the cortex that receives about 94% of blood supply to the kidneys. 94% of blood supply to the kidneys is going to the cortex of the kidneys. Then about 6% is the one that is proceeding to the medulla or the kidneys. So you need to remember that as well. It is surrounded by many capillaries that are very close to the walls. So we said the proximal convoluted tubule is responsible for reabsorption of the majority of the electrolytes. So sodium, potassium, chloride, magnesium. So the reabsorption of those electrolytes is taking place within the proximal convoluted tubule. So, and also reabsorption of glucose, amino acids, and some of these nutrients that are smaller enough to be filtered by the glomerulus, they need to be reabsorbed by the proximal convoluted tubule. So you find that closer to the proximal convoluted tubule, we have peritubular capillaries that are facilitating reabsorption, because whatever is moving from the kidney nephron into the interstitial spaces, they need to move into the cardiovascular system. So nearby the proximal convoluted tubule, we have the capillaries, which are called peritubular capillaries, that are going to facilitate reabsorption. Then they are also going to facilitate secretion. So some substances can move from the capillaries into the interstitial space, then into the lumen or the kidney nephron. That is called secretion, as opposed to reabsorption. Reabsorption is the opposite direction. So substances are moving from the lumen of the kidney nephron into the interstitium, then to the cardiovascular system. Secretion, substances are moving from the cardiovascular system into the interstitium, then into the lumen of the kidney nephron. So I hope you get the difference there between reabsorption and secretion. Approximately 80% of glomerular filtrate is reabsorbed here via selective reabsorption. So you have channels that are responsible for reabsorption of these substances. So 80% of what is being filtered mainly is being reabsorbed here in the proximal convoluted tubule. So it has got a major function with regards to reabsorption of substances in the kidneys. The type of epithelium cells that are found there, they are cubical epithelial cells that will line the tubular walls and have many microvilli on their free surfaces. So they are more like cuboidal cells. So they are cubical cells. They have a lot of those finger-like projections, which are called microvilli. And on these microvilli, okay, they are responsible to increase the surface area for reabsorption and some of the chemical reactions that are also taking place within the kidneys. So they're going to increase the surface area of the wall exposed to the filtrate so that they increase reabsorption and also secretion. The other fact that you need to know, the total surface area of the human proximal tubule cells is about 50 meters squared. So the surface area for the proximal convoluted tubule is 50 meters squared, which means that you're looking at an area of 50 by 50. So just think of the football pitch, now half the football pitch, that's like 50 by 50. So that will be the surface area for the proximal convoluted tubule. So if you combine all the kidney nephrons and then you spread them, the total area that they're going to cover is 50 meters squared. So it gives you an idea to say that there is a lot of reabsorption and secretion taking place within the proximal convoluted tubule. So it's the longest part of the kidney nephron. Functions of the proximal convoluted tubule. So there are 5 major functions that we'll be discussing. Reabsorption of glucose. So the glucose that is being filtered mainly is being reabsorbed within the proximal convoluted tubule. Then hydrogen ion handling. So because the proximal convoluted tubule is responsible for hydrogen ion handling, it means that it's also involved in acid-base balance, because the proximal convoluted tubule can secrete some of the hydrogen ions. So it will have an influence on the body pH. So if the proximal convoluted tubule is secreting a lot of hydrogen ions, then the pH of the urine will be reduced because you have more of the hydrogen ions. An increase in the hydrogen ions, you know, to say that the pH is calculated as the negative logarithm of the hydrogen ions. So if the hydrogen ions concentration is increasing, the pH will be decreasing. If the hydrogen ions concentration is decreasing, the pH will be increasing to be becoming more of alkaline. So if the proximal convoluted tubule, they are secreting a lot of hydrogen ions from the body, the concentration of hydrogen ions in the body will start reducing. That will result into alkalosis. So an increase in pH is called alkalosis. A decrease in pH is called acidosis. So if the proximal convoluted tubule, they are removing a lot of these hydrogen ions, or they are secreting a lot of these hydrogen ions, then the body will suffer from a condition which is called alkalosis. The pH will start increasing. So there are compensatory mechanisms that can come in. So you have respiratory compensatory mechanism or metabolic compensatory mechanisms. So if the kidneys are the ones that are bringing up alkalosis, for instance, the respiratory system can respond by acidosis. So the compensatory mechanism there will be respiratory acidosis. So there will be a balance between alkalosis and acidosis so that the pH is maintained within the normal range. But if your body fails to regulate that pH, then it will result into a disease. That's why disease is failure of the body to maintain these vitals within the normal range. So we have the physiological acceptable values of these parameters. So if your body is unable to maintain those values, then you will fall sick, you become sick. You're unable to do that. That's why you need to go to the hospital so that they can try to resuscitate you, or they can try to bring those vitals or values to normal range so that you become well, okay? So just not to say the proximal convoluted tubule is also involved in hydrogen ion handling. Hence, it plays a major role in acid-base balance as well. It's also responsible for secretion of creatinine. So creatinine secretion is taking place within the proximal convoluted tubule, and secretion of ammonia. So ammonia secretion, creatinine secretion, and secretion of para-aminopuric acid is also taking place within the proximal convoluted tubule. So by now you remember to say, to estimate the glomerular filtration rate, you can use creatinine. But for you to estimate plasma, renal plasma flow, you can use para-aminopuric acid. So such questions come in MCQs. That question that I was asking you to calculate the glomerular filtration rate, I was thinking that everyone was going to get it, but people couldn't even remember the formula for calculating renal plasma flow, of which I told you to say that is the urinary concentration of the substance multiplied by the volume of urine that is collected over a period of 24 hours divided by the plasma concentration of that substance. That is the equation for you to calculate glomerular filtration rate. If you remember that formula, then all the information that you needed was part of that question. So I was wondering why a student couldn't get it right when we discussed in class. Okay. So the other function of the proximal convoluted tubule is responsible for secretion of other substances that are not mentioned on top here. So there are also other minor substances that can be secreted by the proximal convoluted tubule. Okay. So you can see the proximal convoluted tubule that is responsible for reabsorption. So at the center here where you have isoosmotic fluid, this is where the ultrafiltrate is passing. Then you have these cubical cells that are lining the proximal convoluted tubule. So these cells are the ones that will have those proteins that are responsible for reabsorption and secretion. So we say that the proximal convoluted tubule is responsible for reabsorption of sodium, chloride, bicarbonate, potassium, water, glucose, amino acids. So these substances will be reabsorbed. So they will be removed from the lumen, the tubular fluid, into the interstitium, then later on to the peritubular capillaries. Then there is secretion of other substances like hydrogen ions, organic acids, and bases. So you're talking of secretion of creatinine, secretion of paramilipuric acid, and other substances. Okay, so you see that within the proximal convoluted tubule. So handling of glucose, how is this glucose going to be handled by the proximal convoluted tubule? This is very important because even when you are discussing diabetes, you need to know how the kidneys are handling the glucose. Why is it that there will be glucosuria or glycosuria in diabetic patient? Why do we find glucose in the urine of diabetic patient? Does it mean that the kidneys are not reabsorbing much of this glucose or what? So this is where you need to understand that. So glucose and amino acids in the blood are easily filtered by the glomeruli into the renal tubules. So much of this glucose and amino acids, they are so small that they are easily filtered by the glomeruli into the renal tubules. It will become tubular fluid. So glucose is filtered at a rate of. So I hope you've been following the class. Are you getting what I'm saying?
Yes, Doc, we are getting you.
All right. Okay, so the most important thing is to understand what we are discussing here. Remember, learning is not supposed to be one-sided, just a lecturer telling you everything. You also need to spend time researching, studying some of these things so that you update yourselves with the information. So remember, we are looking at the function of the proximal convoluted tubule, of which we said is responsible for reabsorption of a lot of nutrients, amino acids, glucose, electrolytes like sodium, potassium, chloride, magnesium, is also responsible for secretion of hydrogen, paramilipuric acid, creatinine, and other substances. So weak bases, weak acids, they are also being secreted by the proximal convoluted tubule. So in particular here, we are looking at how the proximal convoluted tubule is going to handle the glucose. So the handling of glucose by the proximal convoluted tubule, of which we say that glucose and amino acids in the blood are easily filtered by the glomeruli into the renal tubules because they are so small that they can easily pass through that membrane that is responsible for filtration. Then after glucose has been filtered, okay, so the rate at which glucose is going to be filtered is about 100 milligrams per minute. So every 1 minute, about 100 milligrams of glucose is being filtered by the glomeruli. So these molecules, however, are usually not present in the urine. So they're not going to be present in the urine. So why? It's because almost 100% of glucose has to be reabsorbed so that you have zero concentration of glucose in the urine. So you're not going to see the presence of glucose in the urine or the presence of amino acids. If you do a urinalysis test using the urine dipsticks, so those urine dipsticks, even if you dip those sticks into the urine, you find that you won't find any tracing of glucose because 100% of glucose needs to be reabsorbed. But in case of diabetes, or maybe certain renal diseases that are affecting the filtration and reabsorption or secretion of the kidneys, you can have some amount of glucose in the urine. That is referred to as glucosuria. So it's very common in diabetes. So there is something that is happening to the kidneys for you to have glucose in the urine. So I'm going to discuss that with you, like I've already said. So we proceed. So what is the actual mechanism that is involved in reabsorption of glucose in the proximal convoluted tubule? So there are channels that are responsible for that. So glucose is removed from the urine by secondary active transport. So we have different types of transport mechanism. There is passive transport and active transport. Active transport uses energy. If the proteins that are being used here for active transport, they are hydrolyzing or they are breaking down ATP, they are referred to as primary active transport. If they are using energy that is derived from the movement of other ions, it's called secondary active transport, like in this case that we have here. So I don't know if you have discussed GIT with Dr. Mshavati or Dr. Windi. Otherwise, those transport proteins that are found in the GIT that are responsible for absorption. So in the GIT, you talk of absorption. Here in the kidneys, you talk of reabsorption. So these substances that are being absorbed here, they are being reabsorbed, they are being absorbed for the second time. That's why we refer it to as reabsorption, as compared to absorption that is taking place within the GIT, the gastrointestinal tract system. So absorption of glucose that is taking place in the GIT, the mechanism is somehow similar to what is happening within the kidneys. So we have special epithelium cells that contain special type of channels. So these channels are called sodium glucose 2 carrier proteins or transporter proteins. Sodium glucose 2 carrier that are located on the apical side of the membrane. So apical side of the membrane, you have the sodium glucose 2 channels. Then on the basolateral side of the membrane, we have the GLUT2 channels. The GLUT2 channels are more responsible for facilitated diffusion, but the sodium glucose transporter, these are symporter proteins, meaning that as glucose is moving across the membrane, it can also move with it a glucose molecule. So you have sodium and glucose being transported at the same time. So these are called symporter proteins. If they are moving substances in opposite direction, they are called antipod proteins. So you can have antipod proteins, which are also referred to as exchanger proteins. So it's an exchanger, but this one is a symporter because the direction of movement is into the same direction. So from maybe inside to outside or from outside to inside, it's called a symporter. So glucose and sodium, they're going to bind to sodium-dependent glucose transporter, which are called sodium glucose 2 transporter protein that are located on the apical side of the membrane. Then glucose is carried into the cell as sodium moves down its electrical and chemical gradient. So what you need to understand here is that sodium is moving down the electrochemical gradient. So as sodium is being internalized by the cells down its own electrochemical gradient, it can also facilitate the movement of glucose. So you can see that the movement of glucose here is facilitated by the energy that is derived from the movement of sodium. So it's called secondary active transport. So these epithelium cells, they'll be able to internalize glucose and sodium at the same time. But on the basolateral side of the membrane, you have other types of channels. These are called GLUT2 channels that are involved in facilitated diffusion of glucose. So glucose will start moving now via the GLUT2 channels, the basolateral side of the membrane, into the interstitial, from interstitial into the peritubular capillaries. That will be the overall reabsorption of glucose. So the sodium is then pumped out of the cell into the interstitium by sodium-potassium ATPase pump, but the glucose is transported by the glucose transporter via facilitated diffusion into the interstitial fluid. Then from the interstitial fluid that is located within the interstitium of the cells, it will be reabsorbed into the peritubular capillaries via those openings. Remember, the capillaries, they have those openings which are called the pores or the spaces that can allow movement of certain substances like glucose. So this glucose is just moving via facilitated diffusion from the epithelium cells or the kidney nephrons into the interstitium, then into the peritubular capillaries. if that was complicated, maybe this will simplify your understanding. So we are looking at the proximal convoluted tubule, this portion here. So if we magnify a cross section of the proximal convoluted tubule, you can appreciate the epithelium cells. So these epithelium cells, they are more like cubes, so they are called cubical epithelium cells. They have a lot of finger-like projections, which are called microvilli. So you have these villi-like projections. On these villi-like projections, this is where you have the microvilli that are increasing the surface area for secretion and reabsorption. So here we are discussing the reabsorption of glucose. How is this glucose going to be internalized by these epithelium cells? So the fluid in the tubule lumen, so this is the portion where the fluid is moving after filtration. So this is the lumen at the center of the kidney nephron. So as sodium is moving, as glucose is moving, you have this special type of importer protein. It's called sodium glucose transporter protein 2. So it's when sodium is moving down the electrochemical gradient, it will also move with it glucose. So you can see the internalization of sodium and glucose by the epithelium cells. This is the apical side of the cells. Then on the basolateral side of the cells, you have the GLUT2 channels. The GLUT2 channels will allow the diffusion of glucose. So this diffusion is being facilitated here. So it's called facilitated diffusion of glucose. This glucose is moving into the interstitium. From the interstitium, it will move across the capillary wall to enter the capillaries. So these are the peritubular capillaries. So you can see the overall reabsorption of glucose. On the other side, there is also reabsorption of sodium. So on the basolateral side of the membrane, we said we have sodium potassium ATPase pump that is exchanging sodium for potassium. So it's going to push 3 sodium from inside of the cell to the outside of the cell into the interstitium that will move into the peritubular capillaries. But it's also introducing 2 potassium from the interstitium into the epithelium cells. So this is the internalization or reabsorption of sodium and glucose at the same time. So between the epithelium cells, you have the tight junctions that will prevent paracellular movement of these substances. So remember, you have the fluids that are moving here, you have solutes that are dissolved in these fluids, they are moving within the lumen. So these fluids, they are not able to pass through the tight junctions. So the only way substances can be reabsorbed is via the membrane. So you have the apical side of the membrane, the basolateral side of the membrane that are embedded with a lot of channels responsible for secretion and reabsorption. But in certain location, you can have a bit of paracellular movement whereby the fluids can move in between the cells. So I'll explain later on when we reach there. So that was the mechanism by which glucose is being reabsorbed. So the amount reabsorbed is proportional to the amount filtered. So the amount of glucose that is being reabsorbed is proportional to the amount of glucose that is being filtered. So if 100% of glucose is being filtered, 100% is being reabsorbed. If 70% of glucose is being filtered, 70% will be reabsorbed. That's why I'm saying the amount of glucose that is being reabsorbed is proportional to the amount that is being filtered. Okay, but sometimes there is a point whereby those transporter protein reaches a maximum or transport maximum. So if they are doing 100% job, but there's still a lot of glucose in the lumen, you find that some of it will be remaining there in the lumen. That will result into glycosuria, or the presence of glucose in the urine. So the transport maximum is the maximum rate at which glucose can be reabsorbed from the tubules. So if those transporter channels, they reach a maximum rate of reabsorbing, but there's still a lot of glucose, you'll find that some of this glucose will start appearing in the urine now. So the T maximum, or the transport maximum for glucose, is about 375 milligrams per minute in men and 300 milligrams per minute in women. So it means that in men, if the glomeruli, they are filtering 375 milligrams of glucose per minute. The entire 375 milligrams per minute that is being filtered, it will be reabsorbed. The moment in men they start filtering more than 375, for instance, you start filtering 379, there is some extra amount of glucose that can't be reabsorbed because the transporter protein, they have reached the maximum workload. So the extra glucose won't be reabsorbed. It will appear in the urine. For women, the limit is 300 milligrams per minute. So if they start filtering more than 300 milligrams per minute of glucose, it will start appearing in the urine. So when the T maximum glucose has reached or excreted, it has been exceeded, you find that the amount of glucose in the urine will start increasing. So you don't need to exceed the transportation maximum of these transporter proteins. To summarize that information in a diagram, you can see in this diagram that you are plotting the glucose that is being filtered, then also the glucose that is being reabsorbed and the glucose that is being excreted against the plasma glucose concentration. So in the x-axis, we have the plasma glucose concentration. So starting from 0 to 100, 200, 300, 400, so on and so forth. Then the y-axis, we have the glucose that is being filtered. It's also graded here from 0 all the way to 900 milligrams per minute. So you have this line that is shown in red. So red is representing the filtered load. So how much of glucose is being filtered? The one in blue is showing you the function of those proteins. So the transport maximum or the reabsorption. So the reabsorption that is taking place, that is the function of those proteins that are embedded within the apical side and the basolateral side, reabsorption of glucose. Then the one in green is indicating excretion of glucose. If at all the T maximum has reached. So the moment you exceed the T maximum, you have some amount of glucose that will be excreted. So you have excretion shown in green here. So this is the urine, the concentration of glucose in the urine. So we say that as long as the T maximum is not reached, whatever amount of glucose that is being filtered, it's going to be reabsorbed. So you can see as you are starting from 0 here, 0 as you go to 100, if you filter 100 milligrams of glucose per minute, the reabsorption will also be somewhere around 100 milligrams per minute so that all the glucose is reabsorbed. If you are filtering around 200 milligrams of glucose per minute, what is going to be reabsorbed is 200 milligrams per minute, meaning that everything that has been filtered has been reabsorbed. But as you are approaching the T maximum, the transport maximum for these proteins that are reabsorbing sodium, in women, we say that the T maximum is about 300. So you can see that as you are approaching 300 here, there is some amount of glucose that is appearing in the urine. So you can see now the curve that is shown in green because you are reaching T maximum. So anything above 300 milligrams per minute, then you can see that some of the glucose is being reabsorbed. Those transporter protein, they're doing 100% work. So the maximum they can go is 300 milligrams per minute. The extra amount of glucose, it will be excreted. So you can see that excretion of glucose now will start appearing. Yeah? So if you are filtering about 400, what you are reabsorbing is just about 300, then the extra 100 is going to be excreted. So it's very important for you to understand this diagram because it's just simplifying the reabsorption, secretion, and filtering of glucose, the interaction there. But the most important thing is you need to understand that the proteins that are responsible for reabsorption of glucose, they can reach a maximum workload. Once they reach a maximum workload, then there will be glucose that will be appearing in the urine. So in the case of diabetes, what happens? The concentration of glucose in the blood will increase. So there will be hyperglycemia. So if a patient has got hyperglycemia because of diabetes, there are different types of diabetes. It could be type 1 or type 2, whether the cells that are responsible for production of insulin are destroyed, or maybe there's resistance by the receptor, they are not responding to insulin, that can bring about diabetes. So the cells are not internalizing glucose, the glucose levels in circulation is going to increase, hyperglycemia. So if the concentration of glucose is more than 375 milligrams in circulation, most of it is being filtered. So if, for instance, you are filtering 500 milligrams of glucose per minute, in men, the reabsorption, they will only manage to reabsorb about 375 milligrams per minute. The extra amount will be excreted. So it will appear in the urine because now the transporter protein, they will reach the T maximum, the transport maximum. The extra amount of glucose will appear in the urine. That's why there will be glycosuria. So there will be glycosuria if a patient is diabetic. And you know, to say if there's glucose in the urine, that will simply mean that it will create the osmotic pressure within the lumen or the kidney nephron that will facilitate movement of water from the interstitium into the lumen. So the volume of urine is also going to increase. So there will be glycosuria, there will be polyuria. So the amount of urine that is being produced will also increase, polyuria. So the three clinical signs in diabetes, you know, to say there is polyphagia, polyuria, polydipsia. So I hope you remember that, but that's not the topic that we are discussing for today. Okay, so like I said, in diabetes, there will be glycosuria. What happens is because the transport proteins have reached the T maximum, and the extra glucose that is being filtered there is not going to be reabsorbed because those proteins, they are doing 100% work. So you can only go to 100%. You can't go beyond 100%. So if you reach 100%, you've done what you can. The extra thing can't be reabsorbed. So it will appear in the urine. So in uncontrolled diabetes mellitus, plasma glucose levels are abnormally elevated. So more glucose is filtered than can be reabsorbed. So you find that the amount of glucose that is being filtered is more than what can be reabsorbed. So the urinary excretion of glucose will increase, which is called glucosuria. So you have glucosuria produces an osmotic diuresis resulting into polyuria. So the glucosuria will attract water molecules and then there will be polyuria as well. This is because at a higher concentration, the transport mechanism is saturated and there is no appreciable increment in the amount transported. So once they reach saturation or T maximum, then it means that the extra glucose won't be reabsorbed, like I've already explained. So enough with that, we move on to how the proximal convoluted tubule is handling the hydrogen ions. Like I said, the proximal convoluted tubule is taking part in acid-base balance because it has got a function in handling hydrogen ions. And hydrogen ions, they will have an influence on the pH, whether it's increasing or decreasing. So the cells of the proximal and distal tubules, like the cells of gastric glands, they can also secrete hydrogen ions. So they are responsible for secretion of hydrogen ions. So the channel that is responsible, I'm getting some background noise. Is that a question or what?
Yes, I have a question. In terms of if other than diabetes, if there's sugar in urine, other than diabetes, could it be because of the damage to the proximal tubules, or even a damage to the glomeruli can lead to?
Exactly. So you have
that.
kidney diseases, for instance. So an increase in glucose levels in the urine doesn't necessarily mean that you have a definitive diagnosis of diabetes. No, because there are also other diseases that can result into an increase of glucose in the urine. Like you've already mentioned, if there is damage to the glomeruli or there is damage to the proximal convoluted tubule to the extent that there is no reabsorption of glucose that is taking place there. In as much as what is being filtered is within the normal range, but there are no mechanisms that are responsible for reabsorption because the proximal convoluted tubule are damaged. So there will be glucose in the urine. Okay, but that doesn't necessarily mean that the person is diabetic. It could mean that there is a challenge with the kidneys. So there is chronic renal diseases, there are acute renal diseases, inflammation of the kidneys that can also result into such conditions. But in most cases, when you have an increase in glucose levels in the urine, for you to confirm your diagnosis, you need to do glucose tests. So you can do random glucose tests, random sugar tests. So there are glucometer whereby you can just pick yourself, you squeeze the blood out there, and then you measure the concentration of glucose in the blood. So if the glucose is within the normal range, you know to say it's not diabetic. So you look at other diseases that can bring about that. But if you confirm to say in the urine is glucose, in the blood there's also glucose, it will point to diabetes. Okay, so I hope I've answered that question.
Yes.
All right, so I'll proceed. So the channel that is responsible for hydrogen secretion, they are in the proximal convoluted tubule. So these are called sodium hydrogen exchanger. So sodium hydrogen exchanger is different from a symporter. Remember, for glucose, we say it's sodium, glucose, symporter. But here is an exchanger. So an exchanger is an antiport protein because it's transporting these substances into different direction or opposite direction. So if the direction is opposite, it's called an exchanger or an antiport. The extrusion of sodium from the cells into the interstitium by sodium potassium ATPase lowers the intracellular sodium and causes sodium to enter the cell from the tubular lumen with coupled extrusion of hydrogen ions. So in simple terms, we have sodium potassium ATPase pump that are exchanging sodium for potassium. So they are removing a lot of sodium from the cells. The concentration of sodium within the cells is going to decrease. The concentration of sodium in the interstitium is going to increase, but within the cell it's decreasing because you are pumping much of this sodium to the outside. So if the concentration of sodium is decreasing within the cells, it will facilitate the exchange of sodium and hydrogen ions on the apical side of the membrane. So on the apical side of the membrane, sodium is being internalized by the epithelium cells, then hydrogen is being secreted into the lumen. So this is the exchange between sodium and hydrogen ions. So the hydrogen ions, they come from the intracellular dissociation of carbonic acid. So this carbonic acid is a weak acid that can dissociate into bicarbonate and hydrogen ions. And these hydrogen ions are the ones that are actually being exchanged for sodium. So for each hydrogen ion that is being secreted, at least one sodium and one bicarbonate is going to enter the interstitium or the interstitial fluid. So sometimes it's very difficult to explain from those notes, but diagram is very easy for you to understand. So let me use the diagram for you to understand what is happening there. So I'll use this diagram to explain the handling of hydrogen ions. So let's start. You know to say that substances have been filtered. So you have the tubular fluid that is moving in the lumen of the kidney nephron. As the fluid is moving in the kidney nephron, there are substances that are being reabsorbed, of which we mentioned to say glucose, sodium is being reabsorbed. But these epithelium cells, they also have sodium hydrogen exchanger or antiport protein. What it's doing is that as the sodium is moving, you know, to say after filtration, you have electrolytes like sodium. This sodium is going to be pushed into the cell in exchange for hydrogen ion. So you can see the hydrogen ion that has been exchanged for sodium. This hydrogen ion in the lumen, it can react with bicarbonate to give you carbonic acid. Under the influence of carbonic anhydrase enzyme, the carbonic acid is going to dissociate into water and carbon dioxide. Water and carbon dioxide can easily pass through the phospholipid bilayer to enter the epithelium cells. In the epithelium cells, the carbon dioxide and carbon dioxide and water can react under the influence of carbonic acid to give you carbonic anhydrase to give you carbonic acid. The carbonic acid is going to dissociate into bicarbonates and hydrogen ions. The hydrogen ions are being exchanged for sodium. Then the bicarbonate, they are going to be exchanged for chloride. For instance, you can see this bicarbonate chloride exchanger. So the bicarbonate now is being pushed into the interstitial spaces. And then some of these bicarbonate, they can be transported by sodium bicarbonate syn-porter protein on the apical side of the membrane. So they are being pushed into the interstitial spaces. Then on the basolateral side of the membrane, you also have sodium potassium ATPase pump, of which we know to say that this pump is pumping 3 sodium from the epithelium cells into the interstitium and is pumping 2 potassium from the interstitium into the epithelium cells. So this pump is very important because it's going to reduce the concentration of sodium. That is going to facilitate the exchange of sodium and bicarbonate. Otherwise, if there is a buildup of sodium within the cells, it will be very difficult for this exchanger to exchange sodium for hydrogen ions. So overall, we said for each hydrogen ion that is pumped into the lumen, there is 1 bicarbonate and 1 sodium that is being reabsorbed. That last sentence, you remember? For each hydrogen secreted, 1 sodium and 1 bicarbonate enter the interstitial fluid. So this is what I mean. So for each hydrogen ion that is being exchanged for sodium, at least 1 sodium and 1 bicarbonate is going to be pushed into the interstitium. On top of the sodium hydrogen exchanger, we also have sodium ATPase pump, which are also called proton pump. A proton pump is a pump that can pump hydrogen ions. So you can see that on the apical side, we also have hydrogen ATPase pump that can pump this hydrogen ion into the lumen. So this will have an effect on the pH, because if the cells are losing a lot of these hydrogen ions into the lumen, the concentration of hydrogen ions in the body will be decreasing. So the pH will be increasing. That can bring about acidosis. So if these cells are not secreting a lot of hydrogen ions, there will be a buildup of hydrogen ions in the system or in the body. If there is a buildup of hydrogen ions in the body, that can result into acidosis because the pH will be decreasing. So if you have the kidneys that are bringing in issues with acidosis and alkalosis, the compensatory mechanisms will be provided by the lungs, the respiratory system. That's why if you have metabolic acidosis, the compensatory mechanism will be respiratory alkalosis. If you have metabolic alkalosis, the compensatory mechanism will be respiratory acidosis. The opposite is also true. If you have respiratory acidosis, the compensatory mechanism will be metabolic alkalosis. If you have respiratory alkalosis, the compensatory mechanism will be metabolic acidosis. So they can compensate each other, trying to maintain the pH. And on top of that, you have the buffer systems that can come in to regulate the pH. Okay, so that is handling of the hydrogen ions. What is the fate of hydrogen ion in the urine? The amount of acid secreted depends upon the subsequent events in the tubular urine or tubular urine, of course. So there are also other mechanisms that are taking place beyond the proximal convoluted tubule. So the final fate of hydrogen ions is dependent on other processes that are taking place beyond the proximal convoluted tubule. So some of it can be reabsorbed back, or more hydrogen ions can be secreted into the lumen. Okay, so it will bring about modification of the urine by the other components of the kidney nephron, apart from the proximal convoluted tubule that we're discussing here. So the maximal hydrogen ion gradient against which the transport mechanism can secrete in humans is a pH of 4.5. So if the pH is 4.5, the urine pH is 4.5. This is the limit to which those channels can work. Let me just take you back a bit here. So we have two channels that are secreting hydrogen ions. We said we have the sodium hydrogen exchanger or antiport protein. Then we also have proton pump or hydrogen ATPase pump that are pushing the hydrogen into the lumen. So these pumps, they work well if the pH is 4.5 and above. Once the urine pH becomes 4.5, that's a limiting factor for this protein. So if the pH is below 4.5, they will stop pushing a lot of this hydrogen into the lumen. So that's the limit. So if they reach that limit and then they are not pushing these hydrogen ions, that can bring about acidosis. So the pH needs to be more than 4.5. So what is going to regulate this pH so that it doesn't go beyond 4.5? You have the buffer systems that can take up the hydrogen ions. Okay. So pH 4.5 is the limiting pH for secretion of hydrogen ions. So as long as the urine pH or the tubular fluid pH is below 4.5, those channels, it's very difficult for them to secrete hydrogen ions. I hope you understand. So if there were no buffers that tie up hydrogen in the urine, this pH would be reached rapidly and hydrogen secretion would stop. So because we are saying that the limiting pH is 4.5, within the lumen or the kidney nephron, we have buffer systems that can accept the hydrogen ions so that the pH doesn't go beyond 4.5. So if we didn't have those buffer systems, the pH 4.5 can reach so rapidly that the secretion of hydrogen ions can stop because the limiting pH we are saying is 4.5. So if it's prevented, that can result into acidosis. So it could lead to acidosis if that happens. So what are these buffer systems that are accepting the hydrogen ions? So we have a bicarbonate. The bicarbonate, they can react with hydrogen ions to form carbonic acid, and that carbonic acid can dissociate into carbon dioxide and H2O. The hydrogen ions will decrease and then the pH will start increasing. Then you have the phosphates buffer. So you have hydrogen phosphates that can also accept hydrogen ions to form acids, and these acids, you know, to say that they are weak acids, so the pH is not going to decrease so much because they are accepting those hydrogen ions. So this is called hydrogen phosphates tetraoxide that can accept hydrogen. Then you have ammonia. Ammonia can also accept hydrogen ions to form ammonium. So these are the buffer systems that are accepting the hydrogen ion so that the pH doesn't go beyond, meaning that we have the buffer systems that will ensure that the pH doesn't go below 4.5 because the pH 4.5 is a limiting pH for the secretion of hydrogen ions. So if the pH is going below 4.5, the secretion of hydrogen ions will stop because the channels that are responsible for internalization of sodium and glucose, which are called sodium hydrogen exchanger, which is exchanging sodium with hydrogen, it will stop working. And also the channel, the hydrogen ATPase pump will also stop. So you find that you have buffer systems that can accept the hydrogen ions in the lumen so that it doesn't go beyond 4.5. So these are the ones that I was trying to explain here, though you were not getting what I was saying. So we say that we have these hydrogen ions that are being pushed from the epithelium cells into the lumen by the sodium hydrogen exchanger and sodium ATPase pump. So if the concentration of this hydrogen ion is increasing here, the pH will be decreasing. So if the pH decreases to below 4.5, this secretion will stop. So to make sure that that pH is not going beyond 4.5, you have the buffer systems. So the first one, you have the bicarbonate that can accept the hydrogen to form carbonic acid that will dissociate into carbon dioxide and H2O. Then you have hydrogen phosphate tetraoxide that can also accept hydrogen, giving you weak acid. So this hydrogen ion has been accepted, the concentration will decrease. And then you also have ammonia. Ammonia can accept hydrogen ions to form ammonium. So this ammonium, you know to say that the pH will start increasing because the concentration of hydrogen ions is decreasing as the buffers are accepting the hydrogen ions. That will facilitate the function of sodium hydrogen exchanger and sodium ATPase, hydrogen ATPase pump. Then we have secretion of creatinine. The other function of proximal convoluted tubule is secretion of creatinine. So by now you understand to say creatinine is a byproduct of muscle metabolism. So when you were discussing muscle physiology with Dr. Mchawati, you were taught that the muscle cells, they have energy sources. Those energy sources are the ones that will provide a medium by which muscle can contract or relax. They need energy for them to contract. That energy is in form of ATP. So that ATP can be stored within the muscle cells. You've heard of phosphate creatinine system or phosphocreatinine. So you find that some of the ATP can be stored within the muscle cells. Then if the muscle cells require that energy, it can be converted back into ATP. Okay, so this creatinine is a byproduct from that reaction. So phosphocreatinine can react with ADP to give you back ATP and creatine. Creatine can be converted into creatinine. So it's a byproduct. So if there is muscle damage, you find that the creatinine concentration in circulation will increase. So for instance, you are a wife to somebody, and then for some reason you've been beaten by your husband, and then you go to the hospital to report to say, I was harassed by my husband. Okay, so for us to ascertain that you were beaten by your husband, we need to measure the levels of creatinine. So if you have internal injuries, those internal injuries will cause release of creatinine to the circulation. So if we measure the concentration of creatinine and then we realize to say that this concentration is within the normal range, we'll not to say we are lying. But if the concentration of this creatinine has increased so much, we'll not to say that you have sustained internal injuries. Then we are going to give you a report, a medical report to say you've been abused by your husband. Then you can take it to victim support or wherever you want them to proceed with your case. But what I'm trying to say is that creatinine is a byproduct of muscle metabolism. So it's also freely filtered by the glomerulus. And it's the same creatinine that we were using to estimate the glomerular filtration rate, if you remember. So this creatinine is also actively secreted by the proximal tubule. So there are channels that are responsible for secretion of creatinine. Excuse me. So the knowledge of mechanism of creatinine transport is not well understood. So it's scanty information. So the information is scanty. So I won't give you the mechanism by which creatinine is secreted, but just know to say there are channels that are responsible for secretion of creatinine, but the actual mechanism is not well understood. Then there's also secretion of ammonia. So ammonia secretion by the proximal convoluted tubule. Ammonia is produced in the tubular cells by deamination of amino acids. So deamination of amino acids will result into the production of ammonia. So ammonia is a byproduct of amino acid metabolism, in short. So the proximal tubule has been identified as the principal site of renal ammonia production, as well as an important site of ammonia secretion. So the production and secretion of ammonia is taking place within the proximal convoluted tubule. So the same epithelium cells in the proximal convoluted tubule, they are responsible for deamination of amino acid to produce ammonia, and also they are responsible for secretion of the ammonia after it has been produced by these cubical epithelium cells of the proximal convoluted tubule. So the proximal tubule produce ammonia from glutamine. You know, to say glutamine is amino acids. So deamination of glutamine will result into production of ammonia. So in the tubular fluid, ammonia combines with the secreted hydrogen. I've already told you to say ammonia can function as a buffer that can also react with hydrogen ions to form ammonium. And this ammonium is excreted in the urine. So ammonium is a product that you can see in the urine. So when you are doing urinalysis with Dr. Mshawati, I'm not sure whether you did that lab or not, but when you're doing it, you are using urine dipstick. And those urine dipstick, it was also estimating the levels of nitrates, so nitrate levels. So ammonia is an example. So if you see that the nitrate levels has increased, there's also, you know, to say that even ammonia has also increased in there. Okay. So did you do urinalysis with Dr. Mshawati using those urine dipstick?
Yes, we did.
Yeah, so looking at the levels of nitrate, urobilinogen, glucose, and other substances, pH, then also specific gravity of the urine and stuff like that. So that was done. That's good. Okay, so this is the production of ammonia. So we are saying deamination of amino acids like glutamine. So you can see glutamine, deamination of these amino acids, glutamine will produce ammonia, and this ammonia will be secreted by these cells. So you can see the secretion of ammonia. Then later on, ammonia can react with the hydrogen to give you ammonium that will be excreted in the urine. Okay. How about secretion of paramilipuric acid by the proximal convoluted tubule? So I need to speed up. We are wasting more time here. So the secretion of paramilipuric acid occurs from the peritubular capillaries, blood into the tubular fluid via the carriers in the proximal tubule. So in the proximal tubule, we have carrier for paramilipuric acid that is responsible for secretion of paramilipuric acid. So at low plasma paramilipuric acid concentrations, the rate of secretion increases linearly with the plasma concentration. So what I mean is, just like we discussed for glucose, to say an increase in the filtration of glucose, you have an increase in the reabsorption. So it's the same here. It's also proportional to the amount of paramilipuric acid in the plasma. So if you have an increase, much of it will be secreted as well. So at higher plasma paramilipuric acid concentration, the secretory carriers are saturated. So they can also reach saturation. So once they are saturated, you find that less of paramilipuric acid, not necessarily less, but the channels that are responsible for secretion will reach maximum. So they will secrete as much as they can, but there's just too much of paramilipuric acid that will remain in the circulation. So remember, secretion here is taking place from the peritubular capillaries into the interstitium, from the interstitium into the epithelium cells. So if the paramilipuric acid in the blood is increased so much that the channels that are responsible for secretion have reached a saturation, or they are doing 100% work, but there's still a lot of paramilipuric acid that will remain in circulation. Okay, so that's just like we discussed for glucose, what is happening, but here is secretion rather than reabsorption. So the paramilipuric acid accumulation in the cell is mediated by basolateral membrane organic anion transporter. So we have organic anion transporter that is going to exchange paramilipuric acid with alpha ketoglutarate. So it's exchanging paramilipuric acid with alpha ketoglutarate. So the paramilipuric acid is going to exit the cell passively via the luminal membrane paramilipuric acid anion exchanger. So they also have an exchange on the other side. So from this diagram, maybe you will be able to explain, to understand what I'm trying to explain. So this is a proximal convoluted tubule cell that is responsible for secretion of paramilipuric acid. So you have blood this side, then you have the tubular urine on the other side. So from blood, secretion, things are coming from blood to the epithelium cells, then into the lumen. So you can see that on the basolateral side of the membrane, you have organic anion transporter protein. So this organic anion transporter protein is exchanging paramilipuric acid, which are weak acids, with alpha ketoglutarate. So you can see the alpha ketoglutarate that is coming from amino acids metabolism or protein metabolism, producing the alpha ketoglutarate that will be exchanged with paramilipuric acid. So this paramilipuric acid has moved now from the blood, from the interstitium, into the epithelium cells. Then within the epithelium cells, on the apical side of the membrane, you also have other proteins. So this is called anion paramilipuric acid exchanger. So it's exchanging paramilipuric acid with anion. So you find that this paramilipuric acid now is secreted into the lumen. Then it will be, it's secreted here, then later on, it's going to be excreted by the kidneys to be part of the urine. So there is secretion taking place, then excretion will take place after it has been secreted here. Then you also have reabsorption of other substances. So maybe other substances like organic ions or organic cations. So organic anions and organic cations. The proximal tubule, both the convoluted and the straight portion, secretes a large variety of organic anions and organic cations. So many of these substances are endogenous compounds that are produced within the body, and others exogenous compounds that are coming from outside. So compounds like drugs or toxins, okay? So others are being produced by the body, others can be as a result of what is being introduced to the body from the outside environment. So you have other drugs or toxins that come from outside. So the organic anions are mainly carboxylated. So you find that they are carboxylated, meaning that they will have a carboxyl group attached to them or a sulfur group attached to them. So these carboxylic and also sulfonic acids that will be in their protonated forms, meaning that they'll be carrying a positively charged ion or a negatively charged ion depending on the situation. So these are the ones that are being excreted by the proximal convoluted tubule. So we saw some example here that you also have organic cation transporter. So you have organic cation transporter that is transporting these organic acids, organic substances. Then you have hydrogen ions, organic cation exchanger. So it's going to be exchanged. So overall, the organic substances or compound is being secreted by the proximal convoluted tubule. Okay. There's still a lot of information there. This is just summarizing everything that we've looked at in the proximal convoluted tubule. So the reabsorption of glucose, the exchange of glucose with sodium, the reabsorption of electrolytes, and also the production of ammonia, the excretion of ammonia. So all that. So that's enough for the proximal convoluted tubule. So you need to remember the functions of the proximal convoluted tubule, especially with regard to reabsorption of glucose. Then you should be able to remember the transporter maximum for the channels that are responsible for reabsorption of glucose. When they hit the maximum transport, what happens? So some of the glucose will start appearing in the urine. There will be glucosuria. So if there's glucosuria, it's creating osmotic gradients that will pour a lot of water from the interstitial spaces and also from the epithelium cells into the lumen. So the volume of urine is also going to increase. So you need to be able to explain that with regard to diabetes and other diseases that can bring about glucosuria. Then the handling of hydrogen ions that is involved in acid-base balance, the secretion of hydrogen ions, which channels are responsible for that, the sodium hydrogen exchanger proteins, and also sodium ATPase pump. You need to explain how that is happening, and the function of carbonic anhydrase in secretion of hydrogen ions, the reaction of hydrogen ions with other buffer systems so that the pH doesn't go beyond or below 4.5, which is a limiting pH. Then you also need to understand the secretion of creatinine, though the actual mechanisms is not well understood, but just not to say the proximal convoluted tubule is also responsible for secretion of creatinine. And you know to say it's also responsible for secretion of paramilipuric acid. So you need to understand the channels, of which we said we have alpha glutarate paramilipuric acid exchanger on the basolateral side. Then on the other side, you have anion paramilipuric acid exchanger that bring about secretion of paramilipuric acid. And these channels, they can also reach a transport maximum when they are saturated. So the loop of Henle is the hairpin bend structure. So hairpin bend structure that is going to descend and ascend. So you have the descending limb and the ascending limb of loop of Henle. Main is located within the medulla of the kidney. The descending limb has thin walls, permeable to water, and penetrates deep into the medulla. So the descending has got thin walls and it's penetrating deep into the medulla, is responsible for reabsorption of water. But the ascending limb has thicker and relatively impermeable walls that retains to the cortex. So the descending is more like penetrating deep into the medulla, and then the ascending is moving into the opposite direction. It's going towards the cortex, and it's thicker in terms of the walls of the thick ascending limb of Henle. Then you have they are surrounding surrounding the loop of Henle. So what is surrounding the loop of Henle? You have a network of capillaries. These are called Vasalacta, like you've already explained. Okay, so this is the part of the kidney nephron that I'm now explaining. So we've discussed the proximal convoluted tubule. We are moving down the loop of Henle. So it has got two parts, the descending part, which is thin, and the ascending part. The ascending part, you have thin and thick ascending part of the loop of Henle. So from the descending part, it's very permeable to water. So you can see a movement of water, reabsorption of water. But the ascending parts, which is approaching the cortex, is mainly involved in reabsorption of salts. So you have sodium chloride that is being reabsorbed there. This is the mechanism which I was saying, the countercurrent mechanism that I will discuss later on. Okay. So the loop of Henle works by making the concentration of the interstitial tissues of the medulla hypertonic, greater solute concentration to the filtrate. So the loop of Henle is going to create hypertonic medium in the medulla, as opposed to in the descending part of it, whereby it's very permeable to water, so water can move from the lumen to the interstitium. So just not to say that the loop of Henle is mainly involved in creating the hypertonic or hypertonic medium of the interstitium. So once the medium is hypertonic, you know to say osmosis will be facilitated on the other side of the loop of Henle. So how is this hypertonic concentration created? So the hypertonic concentration is created by actively transporting chloride ions out of the filtrate into the surroundings and also sodium. So once you transport chloride, sodium will also follow. So then sodium ions passively follow. This occurs in the thick part of the ascending limb of loop of Henle. The deeper part of the medulla near the pelvis is the most concentrated and therefore has the lowest water potential. The filtrate at the end of the proximal convoluted tubule entering the loop of Henle is isotonic. So the fluid that is being filtered at the glomerulus mainly is isotonic, meaning that the concentration of electrolytes in that fluid is the same to that of plasma. That's why it's called isotonic. So the process of reabsorption of substances by the proximal convoluted tubule, there's also reabsorption of water. So the fluid that is moving within the proximal convoluted tubule is still isotonic. So as it goes now to the loop of Henle, what happens? So this fluid that is entering the loop of Henle is isotonic in nature, meaning that the concentration of ions in this fluid is the same to that of plasma. So as it descends the loop, it is carried through tissues of increasing solute concentration, and permeable walls of the descending limb enables water to leave the filtrate entering the renal medulla. From there, those are the interstitial spaces of the kidneys. Then it will enter the peritubular capillaries. But remember, the peritubular capillaries of the loop of Henle, they are referred to as Vasalacta. So they will enter the Vasalacta after that. That's the overall reabsorption of water molecules. So this is possible because blood in the Vasalacta is flowing from the deeper, more concentrated regions of the medulla. So its water potential is lower than the filtrate of the adjacent descending limb. So when you say low water potential, it simply means that this fluid is hypertonic. So if it has got low water potential, the fluid is hypertonic, then it's going to facilitate osmosis to take place. So water will move from the hypotonic to hypertonic fluids. So that's why you're going to have an overall reabsorption of water molecules. Okay, so let me just explain from the diagram. Okay, then from the diagram, I'll also explain the countercurrent mechanism due to time. So let me just explain the countercurrent mechanism. Okay, so here is what I'm trying to explain. So you can see that in the thin descending limb of loop of Henle, you have aquaporins 1 protein that are responsible for reabsorption of water. You don't have active sodium transport in the descending limb of Henle because the channels responsible for reabsorption of sodium, they are not there. So mainly it's permeable to water. It's impermeable to solutes. So it doesn't allow movement of solutes, but it's allowing movement of water. Then the ascending limb is responsible for reabsorption of solutes. So you can see that on the other side, there is active pumps that are pumping sodium and chloride from the lumen into the interstitium or from the lumen into the medulla of the kidneys. Actively pumps sodium out of the tubule to the surrounding interstitial fluid. So you have sodium, potassium ATPase pump. On top of that, you have sodium 2-chloride, potassium co-transporter on the luminal side. So on the apical side here, on the apical side of the cells, you have sodium 2-chloride, potassium co-transporter, meaning that these proteins, they are internalizing 1 sodium, 2 chloride, and 1 potassium at the same time. So these electrolytes, they are being reabsorbed. The cells are reabsorbing sodium, chloride, and potassium at the same time. The concentration of these electrolytes will build up in the medulla or in the interstitium. So as they will be built up of solute concentration in the surrounding part of the kidney nephron, it will create the hyperosmolarity. So that hyperosmolarity will cause now osmosis to take place within the descending limb. So in the descending limb where it's permeable to water, water starts moving now from the lumen into the interstitium due to the concentration gradient that has been created by the ascending limb of loop of Henle. So this is called countercurrent mechanism. So the ascending, it's impermeable to water, but it's reabsorbing much of the sodium and chloride. So remember we said as the fluid is approaching the loop of Henle is isotonic or isoosmotic if you want. Isotonic or isoosmotic is one and the same thing. As the fluid is descending in the descending limb of Henle, there is more of reabsorption of water. The concentration will start increasing. The fluid, the tubular fluid becomes hypertonic. Then later on, as the fluid they are ascending in the ascending limb of Henle, you find that there is a lot of reabsorption of solutes. Then the fluid will also become hypotonic or hypoosmotic. So you need to understand all that mechanism. So this is what I'm trying to explain now. This is in the ascending, the thick ascending limb of loop of Henle. The channel that is responsible for internalization of electrolytes there is called sodium 2-chloride potassium. Sodium 2-chloride potassium transporter proteins. So they are able to internalize sodium 2-chloride and potassium at the same time. Then on the basolateral side of the membrane, you have potassium leaky channels, you have sodium potassium co-transporter, then you also have sodium potassium ATPase pump that is exchanging sodium for potassium. Then you also have chloride channels that are responsible for internalization of chloride. So all these electrolytes, they're being pushed into the interstitium. So the concentration of these solutes in the medulla or the kidneys is going to increase so much that it will create the hyperosmolarity in the medulla or the kidneys. That is now going to facilitate osmosis of water because water will start moving now from the descending limb of loop of Henle into the interstitium. Okay, so here it is. So the thin ascending and the thick ascending, that's the one that will contain a lot of sodium 2-chloride potassium channels to create that concentration gradient. Then that concentration gradient will facilitate the thin descending limb to reabsorb a lot of water because it's permeable to water. So one side you have reabsorption of solutes, the other side you have reabsorption of water. But the reabsorption of water is facilitated by the reabsorption of solutes because where the solutes are going, water will follow. So this countercurrent mechanism is going to produce a hyperosmotic renal medullary interstitium. So the interstitium or the medulla in the kidneys are hyperosmotic. In other ways, they are also called hyperosmotic medium because there is a lot of reabsorption of salts that are taking place, especially on the ascending part of the loop of Henle. Then on the descending part of the loop of Henle, that's where you have reabsorption of water that is facilitated by osmosis. So this is basically the mechanism that I want you to understand. So let's just digest the mechanism that are involved in countercurrent mechanism. So from the name itself, countercurrent mechanism, it means that the direction in which the tubular fluid is moving, it's in opposite direction. So you find that in the descending loop of Henle, the movement is downwards. In the ascending, the movement is upwards. So that opposite movement is called countercurrent mechanism. So a countercurrent multiplier system is a mechanism that expands, that will expend energy to create a concentration gradient. So it's going to expend energy to create a concentration gradient because reabsorption of these salts it's against the concentration gradient, so you need energy. So the loop of Henle is going to expend a lot of energy to create the concentration gradient, okay, the hyperosmolarity of the medulla. So the term is derived from the form and the function of the loop of Henle, which consists of two parallel limbs of renal tubules running in the opposite direction, separated by the interstitial space or the renal medulla or medulla. Yes. So this is what I was trying to explain, to say the movement of the fluid is in opposite direction, hence the name countercurrent. There's a diagram that I'll share with you. You'll be able to appreciate that. So the ascending limb is impermeable to water. We say that the ascending is impermeable to water because it doesn't contain aquaporins proteins. The aquaporins proteins are the ones that are being used for the reabsorption of water. So the ascending limb of loop of Henle is impermeable to water because you don't have aquaporins there. And these aquaporins is actually a common transporter protein for water channels. Okay, so most of the cells, they will have aquaporins, but the cells that are found within the ascending limb of Henle, they don't have those aquaporins. That's why this part of the loop of Henle is impermeable to water molecules. So in as much as it's impermeable to water molecules, the ascending limb of loop of Henle is permeable to solutes. So these solutes, these are sodium, chloride, potassium, that are actively transported into the medulla space, making the filtrate hypotonic with higher water potential. What I mean is, as the fluids are moving within the ascending, that ascending limb of loop of Henle is highly permeable to solutes because it's involved in active transportation of these solutes into the interstitium. So as you are pumping these solutes into the interstitium, you are increasing the concentration gradient. So the interstitium becomes hypertonic, but the fluid itself, it will become hypotonic. Why? It's because you are pumping out the solutes from the fluid into the interstitium. So the fluid in the lumen within the loop of Henle, it becomes hypotonic because most of the solutes have been pumped into the interstitium. Okay, so this is very important because the concentration gradient now that has been created by actively transporting solutes into the interstitium is the one that will create the osmotic gradient to facilitate reabsorption of water in the descending limb of Henle. So as the fluids are descending, there is now reabsorption of water on the other side of the loop of Henle. So the interstitium is now salty or hypertonic, and will attract water molecules. So now they will start attracting water molecules. So this constitutes the single effect of countercurrent multiplication process. So let me just explain from the diagram later on for you to enhance your understanding. So the descending limb of loop of Henle is permeable to water, but impermeable to solutes due to the presence of aquaporins 1. So now the descending, okay, so we have the descending and the ascending. So these are the two opposite sides of the loop of Henle. The descending one, it has a lot of aquaporins protein. So it's responsible for reabsorption of water because it's permeable to water. But you'll find that this part of the loop of Henle is impermeable to solutes. So solutes are not moving. What is moving is water. That will bring about increasing the concentration of solutes in the lumen. So as the fluids are moving down the descending limb of loop of Henle, there are water molecules that are being reabsorbed via osmosis. Then the concentration of these solutes will start increasing. So the fluid becomes hypertonic. But as the fluids are ascending, they will become hypotonic because now there will be movement of solutes. There is reabsorption of solutes. So water moves across the tubular wall into the medullary space with a lower water potential. So this is the filtrate that continues to the ascending limb. So in the ascending limb, this is where you have active transportation of solutes. So active transport of these ions from the thick ascending limb, it's going to create an osmotic pressure to draw water from the descending limb into the hyperosmolar medullary space. So the medullary space or the interstitium or the medulla is hyperosmolarity. It's a hyperosmolar, meaning that the concentration of these solids is more. That is going to facilitate now the reabsorption of water in the descending limb of loop of Henle. So as water is being reabsorbed, the fluid, the tubular fluid, becomes hypertonic as well. Okay? So the countercurrent flow within the descending and ascending limb, they're going to increase and also multiply the osmotic gradient between the tubular fluid and the interstitial spaces. That's why it's called countercurrent multiplier effect. So this is just a diagram of the kidney showing the different parts of the kidney. So you have the outermost, which is the cortex. As you go towards the medulla, you can see that there's change in terms of osmolarity. So the cortex is more isotonic or isoosmolar. So as you are moving from the cortex towards the medulla, you can see that there is an increase in the osmolarity of the interstitium. So this osmolarity is created by the function of the loop of Henle. Okay, so I'll explain from the beta diagram for you to appreciate that. So this is what is taking place. So you can see this is a loop of Henle. You have the descending and the ascending. Okay, so the descending, you have thin descending. The ascending, you have two parts. You have the thin ascending and thick ascending. Then we say that the descending is permeable to water, but it's impermeable to solids. So as the fluids are moving from the proximal convoluted tubule towards the loop of Henle, you can see that the tubular fluid here is isotonic. An isotonic fluid simply means that the osmolarity of that fluid is the same compared to that of plasma. If you remember very well, when we were looking at transport across the plasma membrane, we looked at hypertonic, isotonic, and hypotonic. So if the fluid is isotonic, it simply means that it has got the same osmolarity compared to that of plasma. The number of dissolved solutes is the same when you compare this fluid and plasma. So as the fluids are moving from the proximal convoluted tubule, it will bring the isotonic fluid. But now towards the loop of Henle, what is happening here? You can see that the fluid that is being introduced here is about 300 milliosmolar per liter. But as you are going down towards the medulla, you can see there is an increase in the osmolarity. So it has increased from 300 to 600, later on to 900, then down here it's 1,200. So this increase in the osmolarity is due to the reabsorption of water. You can see here that there's a lot of water that is being reabsorbed into the interstitium. As water is moving, the concentration of these solutes is going to increase. Hence, an increase in the osmolarity of the fluid. So the tubular fluid is becoming more of hypertonic, to the extent that if you are at the bottom of the loop of Henle, the osmolarity of the tubular fluid is around 1,200 milliosmol per liter. But now as the fluid is ascending, something else is taking place here. So the ascending limb of loop of Henle, we said it's impermeable to water. So there is no reabsorption of water taking place here. But this is the segment that is responsible to actively reabsorb solutes. So reabsorption of solutes is taking place within the ascending limb of Henle because you have those channels that can use energy to pump solutes against the electrochemical gradient. So you can see now as the fluid are ascending, there is pumping of solutes. You have sodium chloride channels, you have sodium potassium chloride channels that are pumping these solutes into the interstitium. As you are pumping the solutes into the interstitium, water is not following the solutes. So you find that the concentration of these solutes will start decreasing in the lumen. So the fluid, the tubular fluid osmolarity will start decreasing. So as you are ascending, you can see that there is a decrease in the osmolarity of the fluid. So it starts as 1,200. As you are ascending, you're moving those solutes, you are reabsorbing those solutes. It will goes to, let's say, 900. Then from there, it goes to 600. From there, it goes to 300. But you are still reabsorbing these solutes. Then later on, it goes to 100 milliosmolar per liter. So this 100 milliosmolar per liter, the osmolarity is less compared to that of plasma. So this fluid now, as it's leaving the loop of Henle, is more of hypotonic fluid. So the hypotonic fluid, it simply means that the concentration of these solutes are low. That is hypotonic. But the final urine concentration of these solutes is dependent on the functions of other portion of the kidney nephron. Remember, you have the distal convoluted tubule, you have the collecting tubule, you have the collecting ducts that are also responsible for secretion and reabsorption. So depending on those functions, you find that the overall or the final urine that is being produced can be hypotonic or hypertonic, depending on those functions. So there is still some modification that is taking place elsewhere, apart from the loop of Henle. But the countercurrent mechanism, the most important thing that you need to understand is that the ascending part of the loop of Henle is involved in actively reabsorption of solutes. The descending part is just involved in osmosis. But for this osmosis to be facilitated, you need to create the hyperosmolarity in the medulla. So the hyperosmolarity of the renal medulla is created by the ascending limb that is involved in active transportation of solutes. As these solutes are being transported into the interstitium, the concentration of these solutes is going to increase so much that it will create the osmotic gradient that will start facilitating the movement now of water from the descending limb of loop of Henle into the interstitium. That will bring about reabsorption of water. So I hope you understand that. It might sound a bit complicated, but you need to understand. Okay, so the osmolarity, there is osmolarity of the fluid that is moving in the kidney nephron. There is also the osmolarity of the interstitium. Remember, the kidney nephron, they are starting from the cortex, then diving deep into the medulla of the kidneys, then they also go back to the cortex, then later on, they'll start moving towards the medulla again of the kidney nephron. So this kidney nephron is a special structure that is designed to facilitate reabsorption and secretion of certain solutes and substances, as you can see. So the countercurrent mechanism, you can explain it with the osmolarity of the tubular fluid that is moving in the kidney nephron, and also the osmolarity of the interstitium. So the osmolarity of the interstitium, you can see that it's increasing as you are moving from the cortex towards the medulla. So you can see you have the medulla there and the cortex on top. As you are moving towards the cortex to the medulla, there is an increase in the osmolarity. So why is this osmolarity increasing? So you have hypertonic medium here. So why is it increasing? It's simply because on the ascending limb, there are active transportation of solutes into the interstitium. So you can see that there's an increase in the osmolarity. But with regard to the osmolarity of the tubular fluid that is moving in the kidney nephron, as it's introduced to the nephron, to the loop of Henle, we said it's isotonic. So the osmolarity is the same to that of plasma. That's why it's somewhere around 300. So as water is being reabsorbed, the concentration is increasing. So the osmolarity is also increasing. So you can see that the osmolarity increases to about 1,200. Then as you are ascending, you are removing the solutes, the concentration is reducing. So the fluid is becoming more of isotonic, then later on hypotonic. Then there is also modification that is taking place elsewhere. As you can see here, that there is modification that is facilitated by the presence of certain hormones, for instance, ADH, that will cause the collecting tubule and part of the collecting ducts to become permeable to water molecules. So you can see that there is also reabsorption of water that is taking place here in the presence of ADH. Without ADH, this part of the kidney nephron is also impermeable to water molecules. Okay, so the overall components or the overall composition of the urine that is being produced is dependent on all the functions of the kidney nephron, different segments of the kidney nephron. Okay. So still, if you didn't understand the counter current mechanism, I'm just going to summarize it in this diagram now. So if a question comes to say, in essay form, can you explain the counter current mechanism in the kidney nephron? You will need to be able to draw the structure of the loop of Henle and explain the osmolarities within the kidney nephron and also within the Vasalacta. Remember, the Vasalacta, these are peritubular capillaries that are somewhere around the kidney nephron, especially the loop of Henle, if you remember the anatomy. So now let's go through this diagram together so that we summarize. Okay, so if you are not paying attention, now pay attention. So we are saying that the fluid is coming from the proximal convoluted tubule. The function of the proximal convoluted tubule we already discussed. We said it's responsible for reabsorption of most of the solutes. So be it sodium, potassium, chloride, bicarbonates is taking place within the proximal convoluted tubule. Then it's also responsible for reabsorption of amino acids, glucose. It's responsible for secretion of hydrogen ions. It's responsible for secretion of paramilipuric acid and creatinine. So I hope you remember those mechanisms. So some of these channels, they can reach saturation. If they are saturated, it means that they have reached the transport maximum. So you find that if these are channels that are responsible for reabsorption of glucose, some of the glucose will start appearing in the urine because the concentration of glucose has increased so much that those channels, they are doing 100% work, but there is still some amount of glucose that is remaining. Under normal circumstances, we say that all the glucose has to be reabsorbed in the proximal convoluted tubule. So you remember all that. So because in the proximal convoluted tubule, there is reabsorption of electrolytes, there is also reabsorption of water. There is no much change in the osmolarity. So in the proximal convoluted tubule, the osmolarity of the fluid is going to be maintained because it's responsible for reabsorption of solutes and also water at the same time. But as you are approaching the loop of Henle, this is now the counter current mechanism. You have two parts. You have the descending and the ascending. So shown in yellow here, this is a loop of Henle. So you have the descending and the ascending. When you look at the flow of the current here, in the descending is moving downwards, the ascending is moving upwards. That's why it's called counter current mechanism. Counter current mechanism. Even when you compare the Vasalacta, you can see that the movement of blood here, it's moving down and upwards in the opposite direction. It's also counter current mechanism that you appreciate it there. So now let's start. So before we start talking about reabsorption of water, the first thing is you need to create the hyperosmolarity of the interstitium. So the medullary interstitium, you need to create the hyperosmolarity. That is key to reabsorption of water later on. So the hyperosmolarity, we said, is created by the ascending limb loop of Henle. So both the thin and thick ascending limb of loop of Henle, they are involved in creating the osmotic gradient or creating the hyperosmolarity of the interstitium. So you can see there are a lot of sodium chloride channels. We also have sodium chloride potassium channels. So as this sodium is being pumped from the lumen into the interstitium, the concentration of these salts will increase in the interstitium to the extent that they will create the hyperosmolarity. So remember, we defined osmosis as the movement of water molecules from a region of low solute concentration to a region of high solute concentration. So as the ascending is pumping those solutes into the interstitium, the concentration of solutes in the interstitium becomes greater than the concentration of solutes in the lumen or the kidney nephron. So on the descending parts, water will start moving now from low solute concentration to high solute concentration. That's why it's very important for you to understand how this hyperosmolarity is created by the ascending limb of loop of Henle. So the water now will start moving from the lumen. So this is now the descending part, because the descending part is the one that contains aquaporins protein. The aquaporins are responsible for osmosis of water or movement of water molecules. So water molecules now will start moving. So this fluid is at 300 milliosmolar. So as it's going down, water is moving from the lumen into the interstitium. So the osmolarity is increasing. So you can see from 300 it has gone to 600 to 900, then later on to 1200, because water is moving. At the same time, if this water is not being reabsorbed into the Vasalacta, you'll find that this water will start diluting the hyperosmolarity or the interstitium. So if the hyperosmolarity is diluted, it means that the concentration of the solutes will decrease, then it won't facilitate reabsorption. So to maintain the hyperosmolarity, the Vasalacta comes in. So the Vasalacta is the one now that is going to so we are here. So the Vasalacta, you can see that blood is coming from the cortex all the way down, then it's going back to the cortex. So as this blood is coming from the cortex, it comes with blood which is the same osmolarity to that of plasma. So you can see the osmolarity of this blood is at 300. Then as it's going down, there is some amount of solutes that is being reabsorbed into the peritubular capillaries. Then the concentration of these solutes is going to increase. So you can see that the osmolarity within the Vasalacta is also increasing as you are going towards the medulla or the kidney nephron. At the bottom of the Vasalacta, the concentration there is around, or the osmolarity is around 1200. Okay. As now the Vasalacta is ascending back to the cortex, what is happening? Water molecules will start moving now from low water concentration to high water, from low solute concentration to high solute concentration. So this is going to facilitate the overall reabsorption of water. So water is being reabsorbed by the Vasalacta. So the concentration of solutes will be maintained within the interstitium. That will also facilitate more water to be reabsorbed. So this is called the countercurrent mechanism, which is a function of the loop of Henle. Okay, so we proceed. Okay, so now let's look at the urea recycling. Urea recycling. So you know to say that urea is toxic at high levels, but it can be useful in small quantities. So in as much as if there is a lot of accumulation of urea, that could be toxic to the body, but small quantities of urea it's also necessary, because later on you appreciate that certain amount of urea is required to also maintain the hyperosmolarity of the interstitium in the medulla of the kidney nephron. Okay, so the urea recycling causes buildup of high urea in the inner medulla. This helps create the osmotic gradient at the loop of Henle so that water can be reabsorbed. Just like we are talking of solutes that are being pumped, urea is also contributing to that hyperosmolarity of the medulla in the kidney medulla. So here you have the cortex, the outer medulla, the inner medulla. So you will see some amount of urea within the inner medulla that is also creating the osmotic gradient. So urea recycling, you need to understand. So let's start. So let's say 100% of urea is being filtered at the glomeruli. So you can see there's a glomerulus here. Let's say 100% of urea has been filtered. So you can see that you have 100% remaining within the kidney nephron. That will proceed to the proximal convoluted tubule. The proximal convoluted tubule is responsible for reabsorption of urea. So 50% of urea is being reabsorbed in the proximal convoluted tubule. So if 50% has been reabsorbed, what will remain in the lumen will be just 50% because it was 100%, 50% has been reabsorbed. What will remain there is just 50%. So you can see that 50% of urea is remaining. That will proceed to the loop of Henle. Okay, so in the loop of Henle, later on, you can appreciate that in the descending limb of loop of Henle, there is 50% of urea that is being secreted. So if 50% is being secreted, it means that this urea is being pushed into the lumen. So it's being pushed back into the lumen. So if we had 50 plus 50, it goes back to 100. So now as urea is ascending, you have 100% of urea ascending in the distal convoluted tubule. The distal convoluted tubule is responsible for reabsorption of 30%. So 30% of urea is being reabsorbed within the proximal convoluted tubule. What will remain in the lumen here, it will be 70%. So this 70% will proceed to the collecting tubule, to the collecting ducts. So within the inner medullary collecting ducts, they are responsible for reabsorption of 55% of urea. So 55% of urea is being reabsorbed within the collecting ducts. So you can see that what will remain in the lumen now is 15%. So this 15% of urea is the one that is excreted in the urine. So excretion of urea is 15%. If you're doing urine dipsticks, you can see the presence of urea in form of nitrates. The last time we were discussing nitrates. So remember that it will just appear as nitrates. These are urea, 15%. But out of this 55 that is being reabsorbed within the collecting ducts, 50% is the one that is being secreted back into the lumen, and only 5% is being reabsorbed into the Vasalacta or the peritubular capillaries. So the 5% is returning back to the circulation, to the cardiovascular system. So this recycling of urea is called urea recycling. So you don't need to excrete all the urea that you are filtering on the glomeruli. So most of it has to be reabsorbed back into the system. What you are excreting actually is just 15%. So this urea that is being reabsorbed is adding to the hyperosmolarity of the interstitium. That will facilitate the reabsorption of water on the descending limb loop of Henle. So this is called urea recycling. So you know to say at a higher concentration of urea, it's very toxic to the body, but the body has got ways in which it controls the concentration of urea. So it's just going to reabsorb about 85%. The overall 85% of urea is being recycled. Only 15% is excreted in the urine. Okay. So we have less than, yes, aldosterone. So the aldosterone hormone, remember when you're looking at renin-angiotensin-aldosterone system, there's an increase in the release of aldosterone from the juxtaglomerulosa or the zona glomerulosa or the adrenal glands. So they are releasing a lot of aldosterone, and this aldosterone will go to the kidney nephron. So the sites of function, the target site are the principal cells. So it's going to facilitate reabsorption of sodium in exchange for potassium. As the kidneys are reabsorbing much of sodium, water will also follow. So water will be also reabsorbed. Okay. Then we have the intercalated cells that are responsible for reabsorption of potassium ions and secretion of hydrogen ions. So the intercalated cells, they are doing the opposite. So they are exchanging potassium for hydrogen ions. So they're also involved in acid-base balance. So any cell that is capable of secreting hydrogen ions, it's involved in acid-base balance. Okay, so this is what is happening. The principal cells, which are a bit different from the intercalated cells, but I'm showing both here. The principal cells, they are responsible for reabsorption of sodium chloride and water in exchange for potassium, which you can see there. Then the intercalated cells, they are responsible for reabsorption of potassium and bicarbonates in exchange for hydrogen ions. But this part of the kidney nephron is also responsive to antidiuretic hormone. So with antidiuretic hormone, there is increase in the reabsorption of water. So water is going to be reabsorbed with the presence of antidiuretic hormone. The antidiuretic hormone is produced by the posterior pituitary gland. So the posterior pituitary gland, there are two hormones that are being secreted there. We have ADH, which is also called arginine vasopressin. Then we also have the production of oxytocin. From the anterior pituitary gland, there are a lot of hormones that are being produced there. The major hormones, there are 6 there. So you have the production of prolactin, production of growth hormone, the production of thyroid stimulating hormone, the production of gonadotropin releasing hormone, gonadotropin hormone. So there are a lot of hormones that are being produced from the anterior, but the posterior is only responsible for secretion of 2 hormones, ADH and oxytocin. So that oxytocin that is coming from the posterior pituitary gland can go and stimulate the principal cells to exchange sodium for potassium. And also, it's going to facilitate, the ADH actually goes to these cells and facilitate translocation of aquaporins protein. And these aquaporins are the ones that are responsible for reabsorption of water. So it's not going to facilitate reabsorption of sodium in exchange for potassium like aldosterone, but it's going to facilitate the translocation of aquaporins proteins. Moving down to the medullary collecting tubules, what happens there? The medullary collecting ducts reabsorb less than 10% of the filtered water and sodium, because much of this water and sodium has already been reabsorbed from the other segments of the kidney nephron. So they are the final site for processing of the urine. Therefore, they play an extreme important role in determining the final urine output of water and solutes, how much water will be in the urine, because they can also reabsorb some of the water. They can also secrete some of solutes. So the epithelial cells of the collecting ducts are nearly cuboidal in shape with smooth surfaces and relatively few mitochondria. So they have a bit of mitochondria because they're involved in active transport of these solutes and later on reabsorption of water. So you can see the medullary collecting duct that they are responsible for reabsorption of sodium and chloride. Under the influence of ADH, there's reabsorption of water. There is also reabsorption of urea. Okay, so we said 50% of urea is being reabsorbed here. And then there is reabsorption of bicarbonates. There is secretion of hydrogen ions. So it's also involved in acid-base regulation or balance. So the permeability of the medullary collecting ducts to water is controlled by the level of ADH. So with the high levels of ADH, which is antidiuretic hormone, water is reabsorbed into the medullary interstitium, thereby reducing the urine volume and concentrating most of the solutes in the urine. So if there is a lot of ADH, there is a lot of reabsorption of water in this segment of the kidney nephron, then the urine that is going to be produced will be concentrated urine. So highly concentrated urine because much of the water is being reabsorbed. Unlike the cortical collecting tubule, the medullary collecting duct is permeable to urea. So here it's highly permeable to urea. Therefore, some of the tubular urea is reabsorbed into the medullary interstitium, helping to raise the osmolarity in this region of the kidneys and contributing to the kidneys' overall ability to form concentrated urine. So it's also contributing to the hyperosmolality of the medulla, like we've already explained in the urea recycling. So the medullary collecting duct is capable of secreting hydrogen ions against a large concentration gradient, as also occurs in the cortical collecting tubule. So the medullary collecting duct also plays a key role in regulating acid-base balance. I've already mentioned to say, since it's responsible for secretion of hydrogen ions, it's involved in acid-base balance. So the Adh hormone, which is also called arginine vasopressin hormone. So the ADH increases the permeability of the collecting ducts and tubules. So the collecting duct and the tubules, the permeability is going to increase for water. That is going to allow movement of water or reabsorption of water by increasing the translocation of aquaporins protein, which I've already explained. So in the absence of ADH, the collecting tubules and ducts become almost impermeable to water, which prevents significant reabsorption of water and therefore allows extreme loss of water in the urine, also causing extreme dilution of the urine. So without ADH, the urine that a person will be producing will be highly diluted and they'll be producing larger quantities of urine, okay? Because much of the water is not being reabsorbed here without ADH. So higher concentration of ADH have a potent effect on constricting the arterioles throughout the body and therefore increasing the arterial pressure. So the ADH, I said the other name of ADH is arginine vasopressin. So it can go to the blood vessels and bind to arginine vasopressin receptors or ADH receptors and cause vasoconstriction. So if it's causing vasoconstriction, is increasing the total peripheral resistance, blood pressure will increase. It can also cause an increase in venous return because there is venal constriction. So much of the blood is returning to the heart. So if much of the blood is returning to the heart, the venous return is going to increase because much of the blood is coming to the heart via the inferior vena cava and superior vena cava. Then you know to say once the myocardium are stretched more, they're going to contract with a greater force, the cardiac output will increase. If the cardiac output is increasing and also the stroke volume will increase, the blood pressure will increase. So you know to say that ADH is also involved in regulation of blood pressure. An increase in ADH, which is also called vasopressin, it will cause an increase in blood volume. It will cause an increase in vasoconstriction. So blood pressure is going to increase with ADH. So there are two receptors where ADH can go and bind. So when it goes to the blood vessels, it will go and bind to V1 receptor. When it goes to the kidneys, it will go and bind to V2 receptors. So these are two different receptors. So in blood vessels, it will bring about vasoconstriction. That's why the systemic vesicular resistance is increasing with the presence of ADH. And if the resistance is increasing, the work to which the heart has to pump blood against the resistance is increasing. So the workload on the heart is going to increase. That will bring about an increase in blood pressure. Then when it goes to the kidneys, it goes to these cells, then it will bind to V2 receptors. Once it binds to V2 receptors, it will facilitate transcription of messenger RNA, and this messenger RNA is responsible for the production of aquaporins. At the same time, within these cells, the principal cells and intercalated cells, they already have aquaporins that are embedded within the vesicles. So the ADH can go and facilitate the translocation of those aquaporins protein to be embedded within the apical side of the epithelium cells in the kidney nephron to facilitate reabsorption of water. So these are aquaporins proteins. So this is what I'm trying to explain. So we have this hormone which is called ADH that is being produced by the supraoptic and paraventricular nucleus within the brain itself. In the hypothalamus, you have a cluster of nuclei that are responsible for production of hormones. So the nuclei that are responsible for production of ADH and oxytocin, they are called preoptic nucleus and paraventricular nucleus. So not preoptic, but supraoptic. So the supraoptic nucleus and the paraventricular nucleus are responsible for the production of ADH and oxytocin that will be transported and stored within the posterior pituitary gland. So if there's a stimulus that is causing release of ADH, it's going to be released. It will be transported by plasma. So ADH, these are water-soluble hormones. They can just dissolve in water. Then they'll be transported to the kidneys. Within the kidneys, you can see this ADH that has been transported here. It will cross the capillary walls to go into the interstitium because it's water-soluble hormone. It doesn't cross the phospholipid bilayer of the membrane. It just goes and binds to ADH receptors on the plasma membrane of these epithelium cells. So it will trigger a cascade of reaction because it's activating a G protein coupled receptor. So I don't want to go in details explaining the G protein coupled receptors. So they'll bring about translocation of aquaporins. So you can see these vesicles containing aquaporins 2 channels. So they will be translocated on the apical side. Under normal circumstances, you don't see the aquaporins 2. But with the presence of ADH, there is translocation of these aquaporins 2 that will be embedded within the apical side. Then water will start moving from the lumen into the epithelium cells. Then later on, via the aquaporins 3 that are located on the basolateral side of the membrane, the water molecules will move into the interstitium, then overall into the capillaries. So you have overall reabsorption of water with the presence of ADH. So here is binding to V2 receptors as opposed to the blood vessels where it goes and bind to V1 receptors. So this is how ADH is going to facilitate reabsorption of water within the distal convoluted tubule, the collecting duct, and the collecting tubules. So that part of the kidney nephron is impermeable to water, but with ADH, it becomes permeable to water because of those aquaporins. Okay, so if you don't have ADH, you can see that the collecting tubule and the collecting duct is impermeable. Even part of the distal convoluted tubule is impermeable to water molecules. So you're not reabsorbing much of water from this segment. You produce larger volume of diluted urine. So this is the production of larger volume of diluted urine because you don't have ADH. But in the presence of ADH, you can see that there's a lot of reabsorption of water from the distal convoluted tubule to the collecting ducts and also collecting tubules. A lot of water is being reabsorbed, and then the solute concentration is increasing. So you'll be producing small volume of concentrated urine. So whether you're producing concentrated urine or diluted urine is also dependent whether you are producing much of ADH or less. So I hope you understand that. Then the effect of aldosterone. Effect of aldosterone. So let's just discuss aldosterone and then we end here for today. So aldosterone. So this aldosterone is being produced by the zona glomerulosa of the adrenal glands. So if you're looking at the function of renin, aldosterone, renin, angiotensin, aldosterone axis, we say that if there is a decrease in blood volume or plasma volume, the juxtaglomerular apparatus will be stimulated. The macular denser cells will stimulate the granular cells to produce a lot of renin. This renin is an enzyme that will move into circulation. Then there is another precursor that is produced by the liver, which is called angiotensinogen. The angiotensinogen will be converted by renin into angiotensin 1. The angiotensin 1 will be transported or delivered by blood to the lungs, where you find angiotensin converting enzymes. And these enzymes, they will convert angiotensin 1 into angiotensin 2. The angiotensin 2, it has got a lot of functions. So it can cause vasoconstriction, it can stimulate the face center so that you drink a lot of water. It can go to the heart and cause hypertrophy of the heart muscles. You know, it can go to the kidneys and facilitate reabsorption of water there. Then it can go to the adrenal adrenal gland. So the adrenal gland will be stimulated to produce a lot of aldosterone. So this aldosterone, if the concentration of aldosterone has increased, it goes to the kidneys. So what is the function of aldosterone? So aldosterone is a steroid hormone, and you know to say steroid hormones are mainly fat soluble. So for them to be transported in plasma, they need to bind to plasma proteins because they can't easily dissolve in plasma. So they are fat soluble. For them to be transported, they need to dissolve in plasma. So because they don't dissolve so nicely in plasma, there are plasma proteins like the albumin and the globulins that can facilitate the transportation of these steroid hormones to the kidneys. But when they go to the kidneys, because they are fat soluble, they are able to cross a phospholipid bilayer of those epithelium cells to enter the cells and facilitate transcription of certain proteins. So these aldosterone, they go and bind to specific receptors in the cytoplasm of the epithelium cells, which are specific for aldosterone. Then the aldosterone receptor complex is the one now that will move to the nucleus and facilitate transcription of DNA into messenger RNA. And this messenger RNA is responsible for production of proteins. So some of these proteins are channels and others are enzymes that are involved in reabsorption of sodium in exchange for potassium. So some of them are enzymes, others are membrane transport proteins like sodium potassium adenosine triphosphatase that is responsible for breaking down of phosphates to transport these solutes against the concentration gradient. So the aldosterone does not have an immediate effect on sodium transport, but later on you see the effect because it is enhancing the transcription of DNA into messenger RNA. So you can see the aldosterone that is being transported in plasma, bound to plasma proteins, then it can move into the interstitium, the phospholipid bilayer, because they are fat soluble, they cross the phospholipid bilayer to go into the cytoplasm. They will go and bind to aldosterone receptors in the cytoplasm. So the aldosterone receptor complex will move to the nucleus to facilitate transcription of DNA into messenger RNA. The messenger RNA is responsible to cause the ribosomes to translate a lot of proteins into new channels, new pumps, new enzymes that are involved in reabsorption of sodium in exchange for potassium. So you can see that sodium is being reabsorbed in exchange for potassium. On the basolateral side of the membrane, you have sodium potassium ATPase pump that is also pumping sodium out of the cells and potassium into the cells. So there is overall reabsorption of sodium. So where sodium goes, water follows. So even water will start moving towards sodium. So there will be overall reabsorption of sodium and water under the influence of aldosterone, which is different from ADH. Okay, so I think too much of information is not good. Too much of data is not good. So for today, I think we end here. The next time we meet to look at the process of urinating. Okay, so it's a reflex. The micturition reflex, how is urination triggered in a normal person who's not sick? Then if there are these reflexes, they are compromised, what happens? Maybe if there is spinal cord injury, maybe at the level of the sacral region where you have centers that are responsible for urination and defecation, what happens if you are injured somewhere around the sacral region here? So that we'll discuss next time. So for now, we've looked at the function of the kidney nephron, starting from the glomerulus all the way to the collecting tubules and collecting ducts. So those are the major functions of the kidneys: reabsorption, secretion, and excretion of waste products. There are different channels responsible for that.